Previously, we discussed single sequencing, which rely on capturing signals when specific bases are added to a growing DNA strain. Today, advances in technology have made DNA sequencing much more efficient and high throughput, especially with methods like sequencing by synthesis. In the modern approach, DNA is first fragmented using the enzyme. Then the resulting fragment are then hybridized to a surface containing many adapters, short, non-sequenced that serve as anchors. These adapters allow the fragment to bind to a specific location or length on the sequencing platform. Once attached, the fragments undergo a process similar to the PCR, where they are amplified to create a cluster of identical DNA molecules. This is achieved by the fragment binding to an adapter, then folding over to attach to the nearby adapter, forming a bridge. This bridge amplification results in many copies of each DNA fragment, increasing the signal for a subsequent sequencing step. Amplifying a single DNA fragment into many identical copies ensures that the signal is strong and reliable for detection. In the sequencing step, special fluorescently labeled nucleotides are added. When a nucleotide is incorporated into a new DNA strain, it emits a light signal without terminating the chain. Sophisticated imaging systems capture this light signal in the real time for millions of clusters simultaneously. By analyzing the sequence of emitted signals, computers can accurately reconstruct the original DNA sequence. Sequencing many identical copies of each fragment, much like repeating an experiment multiple times, greatly increases the reliability of the resulting sequencing data. Next Generation Sequencing (NGS) has revolutionized DNA analysis by enabling the rapid and affordable parallel sequencing of millions of DNA fragments. Several core technologies drive this field. Platforms based on sequencing by synthesis (SPS), such as Illumine and MGI, detect each nucleotide as incorporated into a growing DNA strain, producing highly accurate short reads. The unturned technology, in contrast, also sequenced by synthesis, but detects the release of hydrogen ion rather than the light of fluorescence, offer an alternative approach for short-read sequencing. Beyond SPS, there are long-read platforms like Oxford Nanopore and Pacific Bioscience (PacBio) that use fundamentally different principles. Oxford Nanopore technology reads individual long DNA molecules directly by measuring the change in electrical current as a nucleotide pass through a tiny nanopore, where PacBio's single molecule real-time (SMRT) sequencing uses light detection to monitor the addition of nucleotides to a single DNA molecule in the real time. Both long-read technology can generate reads tens of thousands of pages long, which is invaluable for resolving repetitive and complex genomic regions. After sequencing, sophisticated algorithms assemble the millions or billions of the short and long reads into complete genome by finding overlaps and piecing the fragments together, much like reconstructing a scrapbook. Today, best practice often involves combining the high accuracy of short-read platforms like Illumine or Antorrent with the long-range information from long-read technology like Oxford Nanopore or PacBio. This hybrid approach results in genome assemblies that are both highly accurate and capable of spanning complex or repetitive regions, overcoming limitations of either method alone. For example, an initial rough genome may be constructed using short reads, then validated and refined using long reads, which can tolerate a few mismatches but have confirmed the overall sequence structure.